Hello, everyone. Welcome to our another session, and I'm going to discuss to you the protein, which is the third type of biomolecule. And this presentation is taken from the General Organic and Biological Chemistry of Stephen Stocker. Proteins are mainly present in the meat and dairy products, and proteins are naturally occurring unbranched polymer in which the monomer units are the amino acids. I would like again to reiterate that the monomer unit of protein is amino acids. And proteins are most abundant in molecules in the cells after the water. Proteins account for about 15% of a cell's overall mass. And proteins mainly composed of these elements: carbon, hydrogen, nitrogen, oxygen, and sulfur. And the average nitrogen content of a protein is 15.4% by mass. And there are some other proteins that are present in their structure: the iron, the phosphorus, and other metals in some specialized proteins. In general definition of proteins, it is said to be the naturally occurring and branched polymer with monomer units that are called as your amino acids. In specific definition, a protein is a peptide in which at least 40 amino acid residues are present. The term polypeptide there and the protein are often used interchangeably to describe a protein. And several proteins with greater than 10,000 amino acid residues are known already, and common proteins contain 400 to 500 amino acid residues, and there are some small proteins that contain 40 to 100 amino acid residues. And there are more than one polypeptide chain may be present in a protein. We call it monomeric if it contains one polypeptide chain, and we call it multimeric if it contains two or more polypeptide chains. Proteins are classified based on their chemical composition. It's either a simple protein or a conjugated protein, or it's a complex proteins. First, let's focus our attention on the simple proteins. Simple proteins are proteins in which only amino acid residues are present, and more than one protein subunit may be present, but all subunits contain only amino acid. This includes albuminoids, the albumins, the globulins, and histones. When we say albuminoids, these are present in our keratin of the skin, hair, nails, the collagen in cartilage. If we say albumins, it is present in egg albumin and serum albumin. Globulins that is present in our antibodies, histones that are present in the chromatin in chromosomes. Another classification of protein is the conjugated or complex proteins. These are proteins that has one or more non-amino acid entities, or we call it prosthetic groups, that are present in its structure. Prosthetic group may be organic or inorganic. To elaborate, when we say organic, so for prosthetic group contains organic because the prosthetic group contains carbon in its structure, while inorganic there is no presence of carbon. We have here several prosthetic groups: the heme unit, the lipid, the carbohydrate, the phosphate group, the nucleic acid, and the metal ion. So these actually belongs in a different classes, okay? And for example, we have the class hemoproteins, the heme unit. The specific example is the hemoglobin and the myoglobin. So the function of these carrier of oxygen in the blood, your hemoglobin, and the myoglobin is the oxygen binder in the muscle. Another, we have the lipoproteins, which the prosthetic group is lipid. So the specific examples here are the LDL and the high or HDL, the low density lipoprotein, the high density lipoprotein. And these are actually lipid carrier. Okay, another, we have the glycoproteins, which are carbohydrate, and specific examples are the gamma globulin, the mucin, and the interferon. So your globulin is the antibody, the mucin is the lubricant in mucous secretions, and your interferon is antiviral protection. Another, we have the phosphoproteins. So the prosthetic group here is the phosphate group, and the specific example is the glycogen phosphorylase, that is an enzyme in glycogen phosphorylation. Another, we have the nucleoproteins. The prosthetic group is nucleic acid, and the specific example is the ribosomes and viruses. So the functions of these examples, the ribosome is the site of protein synthesis in the cell, and the virus self-replicating infectious complex. Lastly, we have the metalloproteins. From the term, prosthetic group is the metal ion, and the specific example is the iron, ferritin, and the zinc alcohol dihydrogenase. So the function of your iron, ferritin is the storage and complex for iron. I mean storage complex for iron. And the zinc alcohol dihydrogenase is an enzyme in alcohol oxidation. Another classification of protein is based on its shape. So one classification of your protein is a fibrous protein that includes the alpha keratin and the collagen. So these are the polypeptide chains are arranged in long strands or sheets and have long rod shape or string-like molecules that can intertwine with one another and will form a strong fiber, and these are usually water insoluble. So the function of these fibrous proteins that includes the alpha keratin and the collagen is for structural purposes. Another classification of protein based on its shape is a globular protein. This includes the myoglobin and the hemoglobin. So the polypeptide chains are folded into spherical or globular shapes. And the non-polar amino acids are in the interior, and the polar amino acids are on the surface. Okay? And we already discussed the non-polar and the polar ends of proteins last time. Okay? So water soluble, these are water soluble, which allows them to travel through the blood and other body fluids to sites where their activity is needed. And the functions is said to be dynamic. Why is it dynamic? Because these are the one that carries oxygen in the body. The hemoglobin that carries oxygen in the blood, and the myoglobin in the muscle. In this table, it shows the common fibrous and globular proteins and their occurrence and function. So we have here the fibrous proteins, which are insoluble. So these includes the keratins, the collagens, elastins, myosins, and fibrin. So keratins are found in wool, feathers, hooves, silk, and fingernails. The collagen found in tendons, bone, and other connective tissue. The elastins found in blood vessels and ligaments. Myosins found in muscle tissue, and the fibrin is found in the blood clots. On the other hand, globular proteins, which are soluble, includes the insulin, myoglobin, hemoglobin, transferrin, and immunoglobulins. Your insulin, it is a regulatory hormone for controlling glucose metabolism, And myoglobin involves in oxygen storage in muscles. Your hemoglobin involved in oxygen transport in the blood. The transferrin involved in iron transport in blood, and the immunoglobulins involved in immune system responses. Another classification of protein is based on its function, because proteins play a crucial role in most biochemical processes, and the diversity of functions exhibited by proteins far exceeds the role of other biochemical molecules. So the functional versatility of proteins stems from the ability to bind small molecules specifically and strongly, the ability to bind other proteins and form fiber-like structures, and the ability to integrate it into the cell membranes. One example is that your glycoproteins that is present in the cell membrane, that is very important in the cell-to-cell recognition. As I said earlier, we categorize proteins also based on their function. So there are several major categories of proteins based on their function. We have the catalytic proteins. Your catalytic proteins, these are enzymes that are best known to be their catalytic role. And enzymes are almost every chemical reaction in the body is driven by that particular enzyme, but with a specific enzyme. Another category is a defense proteins that includes immunoglobulins and antibodies. These are central to functioning of the body's immune system, especially like for example, we develop antibodies after we have the vaccination shots, the first, the second dose, and the booster against COVID-19. So we develop antibodies, and these are actually proteins. Another is transport proteins. When we say transport proteins, these are proteins that bind small biomolecules, example, oxygen and other ligands, and transport them to other locations in the body and release them on demand. Another type or another category is your messenger proteins. These messenger proteins transit signals to coordinate biochemical processes between different cells, tissues, and organs. Example are your insulin and glucagon, which regulate carbohydrate metabolism, and another is your human growth hormone that regulates body growth. Another type or another category is the contractile proteins. These are necessary for all forms of movement. And one example is the actin and myosin that the muscle contain. And these are filament-like contractile proteins. Okay, so these are myosin and actin. For human reproduction, it depends on the movement of the sperm, which is possible because of the contractile proteins present in the flagella of the sperm. Another is your structural proteins. The structural protein usually confers stiffness and rigidity. So we have different types. One is your collagen and one is your keratin. Your collagen is a component of our cartilage, and our keratin, this usually gives mechanical strength as well as a protective covering to our hair, fingernails, feathers of birds and hooves, and other structures of animals. Another, we have transmembrane proteins. Transmembrane proteins, these are actually a span in the span of a cell membrane. It is usually help control the movement of small molecules and ions. They have channels which help molecules to enter and exit the cell, and the transport is very selective. That means they allow passage of one type of molecule or ion only. Okay? So that is why these are very important in the very important structure present in our cell membrane. This adds our cell membrane as selectively permeable membrane. Another category is storage proteins. Storage proteins, these are actually bind or store small molecules. One example is your ferritin, which is an iron storage protein that saves iron for use in the biosynthesis of a new hemoglobin molecules. Another is the myoglobin, which an oxygen storage protein present in the muscle. Another category is regulatory proteins. Regulatory proteins often found embedded in the exterior surface of the cell, and it acts as sites for receptor molecules. These these proteins often bind to the enzyme, which is the catalytic protein, and thereby turning them on and off, and that is why it controls the enzymatic activity or enzymatic action. Another category is the nutrient protein. This nutrient protein particularly important in the early stage of life from embryo to infant. One example is your casein, which is present in the milk, and albumin, which is present in the egg white, which are said to be the nutrient proteins. Milk also provide immunological protection for mammalian young because it contains a lot of antibodies that are being passed by the mother to the infant through breastfeeding. Now, as I mentioned in our discussion, the amino acids are the building blocks of proteins. And what are these amino acids? Amino acids, it's an organic compound that contains both an amino or amine group, which is NH2, and a carboxyl group, which is C double bond OH, that are attached to same carbon atom. In its structure, so the position of carbon atom is alpha, while the NH2 or the amine group is attached to the alpha carbon, while the the COOH group or the carboxyl group is attached at the alpha carbon also. So the R group or the side chain varies in size, in shape, in its charge, in its acidity, and functional groups that are present, the hydrogen bonding ability, and the chemical reactivity. Greater than 700 amino acids are known, and based on common R groups, there are 20 standard amino acids. Let us try to look into this illustration or these illustrations. Let's focus in this first. So this is the alpha carbon where the amine or the amino group is attached and the carboxyl group is also attached. So here the R is the side chain, and usually there are 20 standard amino acids based on their R groups. Okay, so this one is the proline, which the COH and the NH group or the carboxyl group and the amino group is attached to the alpha carbon. So this is actually the R group already or the side chain. Okay, how about naming of these amino acids? So common names assigned to amino acids are currently used. The three-letter abbreviation that is widely used for naming. So the first letter of amino acid name is compulsory and capitalized, followed by the next two letters not capitalized except in the case of asparagine, glutamine, and tryptophan. So one letter symbol, it is commonly used for comparing amino acid sequences of proteins, and these are usually the first letter of the name. For example, the asparagine, the first letter is the A. So this is the one letter symbol of asparagine, the A. For the glutamine, it's G. The tryptophan, it's T. Okay? When more than one amino acid has the same letter, the most abundant amino acid gets the first letter. That's how we name amino acids. Another thing that we need to understand or to remember is that all amino acids differ from one another by their R groups. And there are 20 common standard amino acids. These standard amino acids are divided into four groups based on the properties of the R groups. We have the non-polar amino acids where the R groups are non-polar. Okay? When we say non-polar amino acids, the R groups are non-polar. And such amino acids are hydrophobic, that means water-fearing, and these makes them insoluble in water. 8 of the 20 standard amino acids are said to be the non-polar amino acid, and when present in proteins, they are located in the interior of protein where there is no polarity because they are said to be hydrophobic or water fearing. So these are the examples of non-polar amino acids. This includes the glycine, the alanine, the valine, the leucine, the isoleucine, the proline, the phenylalanine, the methionine, and the tryptophan. Okay? So these are, again, the non-polar amino acids. Another type is your polar amino acids, where the R groups are said to be polar. So we have three types of polar amino acids. This includes polar neutral, polar acidic, and polar basic. When we say polar neutral, it contains polar but neutral side chains, and this includes 7 amino acids that belongs to this category. Another is the polar acidic. This contains carboxyl group as part of the chain, and we have 2 amino acids belong to this category. Another one is your polar basic. It contains amino acid as part of the side chain. There are 2 amino acids belongs to this category. In this illustration, here are the polar neutral amino acids. The serine, the cysteine, the threonine, the threonine, the asparagine, the glutamine, and the tyrosine. These two are polar acidic amino acids, aspartic acid, and the glutamic acid. These three are the polar basic amino acids, the histidine, the lysine, and the arginine. We have another which is non-standard amino acids. These non-standard amino acids are called to be the derived amino acids, and they are usually formed by an enzymatic facilitated reaction in a common amino acid after that amino acid has been incorporated into a protein structure. Examples of your derived amino acids are your cysteine, your desmosine, and the isodesmosine that are found in the elastrin, hydroxyproline, and hydroxylysine found in the collagen, which is the gamma carboxyglutamate found in the prothrombin. Now, let's talk about essential amino acids. What are these essential amino acids? These essential amino acids, a standard amino acid that are needed for protein synthesis that must be obtained from dietary sources. And adequate amounts cannot be synthesized in human body. That is why we need to obtain it from our dietary sources. So there are 9 of the 20 standard amino acids are considered essential, and these includes the arginine that is required for growth in children and is not essential for adults. We have the histidine, the isoleucine, the leucine, the lysine, the methionine, the phenylalanine, the threonine, the tryptophan, and the valine. Before we call a protein as a protein itself, these monomers of amino acids must join together. And what joins these amino acids? These are peptide bonds. Okay? So under proper conditions, amino acids can bond together to produce an unbranched chain of amino acids. And they are joined together by a peptide bond. So the reaction is between amino acid of one amino acid and carboxyl group of one amino acid, meaning the the reaction in forming the peptide bond is the is between the amino group of one amino acid and the carboxyl group of one amino acid. So meaning this carboxyl group will attach to this amino group of your another amino acid, forming this peptide bond here. Okay? So the length of the amino acid can vary from a few amino acids to hundreds of amino acids. Such a chain of covalently linked amino acids is called the peptide. So, the covalent bonds between amino acids in a peptide are called peptide bonds. And these are already amide. So, what is the nature of that peptide bond? By convention, the structure of peptides is represented beginning with the amino acid whose amino group is free, or the N-terminal end. The other end contains a free carboxyl group, and it is called as the C-terminal end. Okay? The N-terminal and the C-terminal. The amino acids are added to a peptide by forming peptide bonds with the C-terminal amino acids. So meaning the head there is the N-terminal end, and the addition of amino acids or forming of peptide bonds is in the C-terminal end. Why is it C-terminal? Because it is in the carboxyl group, the C. How about the N-terminal? Because it's in the amino group. That is why N-terminal end. Okay? Also, this is the N-terminal end. This is the C-terminal end because of the carboxyl, because of the amino group. Okay? So where is the peptide bond? Here is the peptide bond. Another peptide bond. Okay? The bonding of the carbon to nitrogen in the peptide linkage has partial double bond character that makes it rigid and prevents the adjacent groups from rotating freely. That means the peptide bond is planar and the two adjacent alpha carbons lie trans to it. So the H or the hydrogen of the amide nitrogen is also trans to the oxygen of the carbonyl group. And the almost of the peptide bonds in the proteins are planar and have a transfiguration. Okay? How about the peptide nomenclature? The C-terminal amino acid, the residue skips its full amino acid name, and all the other amino acid residues have names that end in YL. The YL suffix replaces the I and E or the IC or the IC or the acid ending of the amino acid name, except for tryptophan, for which the YL is added to the name. So the amino acid naming sequence begins at the N-terminal amino acid residue. For example, here, the alanine, the leucine, the glycine has the IUPAC name of alanyl leucyl glycine. We have also biochemically important small peptides, and many relatively small peptides are biochemically active. These includes hormones, neurotransmitters, antioxidants, and artificial sweeteners. One is your small peptide hormones. Your small peptide hormones, these are best known as peptide hormones, oxytocin and vasopressin. So it produced by the pituitary gland, and these are non-peptide or 9 amino acid residues, with 6 of the residues held in the form of a loop by a disulfide. Here, the disulfide bond formed between two cysteines residue. Okay? So this is an example of your small peptide hormone, which is the oxytocin and vasopressin. Another, we have the enkephalins. Their enkephalins are pentapeptide neurotransmitters produced by the brain and bind receptors within the brain and usually help reduce pain. This enkephalins, best known as your meth enkephalin and leu enkephalin. Another, we have small peptide antioxidants. Another is glutathione. This is actually a tripeptide which is present in high levels in most cells. And these are usually small peptide antioxidants. The glutathione is a regulator of oxidation reduction reactions and is said to be an antioxidant and usually protects the cellular contents from oxidizing agents such as peroxides and superoxides. These are highly reactive form of oxygen often generated within the cell in the response to bacterial invasion. And what protects it? Protects us, protects our cell, it's glutathione. Usually, its unusual structure feature is glycine is bonded to the cysteine through the side chain carboxyl group. This one, in this structure. Another, we have small peptide artificial sweeteners. One is your aspartame. This is dipeptide that are sold under the trade names of Equal and NutraSweet. And this is usually 180 times as sweeter or sweet as sucrose. This is the structure of your aspartame. This is a combination of the aspartyl, phenylalanine, and the methyl ester, usually present in our um, commercialized products that makes it sweeter compared to sucrose. Now let's talk on the structures of proteins, and it includes primary, secondary, tertiary, and quaternary structure of proteins. When we say primary structure of protein is usually the order in which amino acid are linked together in a protein by a peptide bond. So the backbone of the protein molecule, this is your primary structure. And every protein has its own unique amino acid sequence. This is according to the Friedrich Sanger sequence and determine the primary structure for the first protein, which is the insulin. The structure of your proteins or the connection of the different amino acids in your protein is very crucial. So the order in which the amino acid residues of a peptide molecule are linked is the amino acid sequence of the molecule. So differences in the chemical and physiologic properties of peptides results from differences in the amino acid sequences. Let's say, for example, we have the bradykinin and the boguskinin or kinine. So usually this is the sequence of amino acids. So they have different different sequence of amino acids that makes them different from each other. So your bradykinin or kinine is partly responsible for triggering the pain, welt formation, movement of smooth muscle, and lowering of blood pressure. On the other hand, the boguskinin is completely inactive. That is why the name bogus or false. Another example here is the the condition of our red blood cells. So the amino acid sequence for the normal red blood cell is this. While there are problems like the sickle cell anemia and Georgetown anemia, there is a difference of in their sequences compared to the normal. In this portion here, this is the normal, valine here ang sa imo sickle cell. Okay? Also, the lysine here in the Georgetown anemia. As you can see here, this is the normal red blood cells, and it is compared to the sickle cell red blood cell. So this is the structure. So this leads to a severe anemia for those persons with this condition. In sequencing a protein, we have three steps. We have the hydrolysis, the identification of the products of hydrolysis, and fitting the pieces together as a jigsaw puzzle. So first we have hydrolysis. This is usually effected by the acid or the alkali or an enzyme. So for example, we have the acid hydrolysis. So heating in presence of the 6 molar hydrochloric acid at 110 degrees Celsius and 10 to 100 amino acids destroys. Okay, so um, this is the protein sequence. Okay, I mean the amino acid sequences. So will undergo heat in the presence of the of heat here, it will produce these things. Okay, the alanine, the glycine, and the cysteine. How about the alkaline hydrolysis? In alkaline hydrolysis, heating in the presence of 4 molar sodium hydroxide and does not damage the tryptophan but destroys a lot more amino acid. Next, we have the enzymatic hydrolysis that is used usually of proteases and peptidases. Another, we have enzymatic hydrolysis that includes the exopeptidases and endopeptidases. So when we say exopeptidases, this actually cleaves external peptide bonds. That is why it is said to be exo. So we have two types. We have aminopeptidase and your carboxypeptidase. Your aminopeptidase sequentially cleave peptides beginning at the N terminal. That's why amino. We have also the carboxypeptidase. The carboxypeptidase, it's actually it cleaves peptide bonds beginning at the C terminal end. That is why it's carboxypeptidase. Another, we have endopeptidases. Endopeptidases, it cleaves internal peptide bonds. That is why it is said to be endo, meaning susunod. So we have the trypsin, the chymotrypsin, and the pepsin. These things are very important in digestion of proteins in the in our digestive system. So the trypsin cleaves peptide bonds at the carboxyl end of two strongly basic A's, the arginine and the lysine. Another, we have the chymotrypsin or chymotrypsin. It cleaves peptide bonds at the carbonyl end of the three aromatic A's. So another, we have the pepsin that cleaves peptide bonds at the amino end of the three aromatic A's or amino acids. Okay? AA, that means amino acid. Next, we have the secondary structure of proteins. Your secondary structure of proteins, the arrangement of atoms of peptide backbone in space, is this one. Okay? Your secondary structure is result of hydrogen bonding. So that's why they have a alpha helix and the beta plated sheet. So the peptide linkages are essentially planar. That is why it allows only two possible arrangement for the peptide backbone for the following reasons. We have two amino acid linked through a peptide bond. Six atoms lie in the same plane. The planar peptide linkage structure has considerably rigidity and therefore rotation of the groups about the carbon and nitrogen bond is hindered. So the cis and the trans isomerism is possible about the carbon-nitrogen bond, and the trans isomer is the preferred orientation. So the only bond responsible for the secondary structure of protein is the hydrogen bonding between peptide bonds. So the C double bond O of one peptide group and the N or nitrogen hydrogen bonding of another peptide linkage farther along the backbone. So the two most common types of our secondary structure of protein is your alpha helix and the beta plated sheet or the B plated sheet or these two here. When we say alpha helix, it is a single protein chain that adopts a shape that resembles a coiled spring. So this one. Okay, that's why gintawag siya nga helix. So the hydrogen bonding between amino acids there with the same chain is said to be intramolecular hydrogen bonding. They are coiled in helical spring and the R groups stay outside of the helix and not enough room for them to stay inside because they not have the enough room for to stay for them in the inside. So the helix is so tightly wound and the space in the center is too small for the solvent molecules to enter. Okay? So this is actually the top view of an alpha helix showing the amino acid chains, or the R group is situated away from the long axis of the helix, or these are actually present outside the helix. On the other hand, we have the beta plated, or yeah, helix or sheets, completely extended protein chain segments in the same of different molecules governed by intermolecular or intramolecular hydrogen bonding. So the H, or the hydrogen bonding between different parts of the single chain is the intramolecular hydrogen bonding. The side chains below or above the axis. Okay? And usually they have the U-turn structure most frequently encountered. So this is the U-turn. So naga liko-liko siya. So it starts to wind. In addition, the beta-pleated sheets completely extend protein chain segments in same or different molecules. The hydrogen between different chains are said to be intermolecular bonding. They are said to be parallel and anti-parallel. When we say parallel, the chains run in the same direction. When we say anti-parallel, chains run in opposite direction. And these are usually more stable because of fully collinear hydrogen bonding. So mostly, the anti-parallel is the most stable compared to the more stable compared to the parallel beta-plated sheets. Next structure of the protein is your tertiary structure. Overall, this is a three-dimensional shape of protein that results from the interactions between the amino acid side chains, or what we call as the R groups, that are widely separated from each other. Usually, the tertiary proteins or tertiary structure of protein defines the biological functions of proteins. Proteins may have, in general, either of the two forms of tertiary structures, which I've mentioned in our previous slides, the fibrous and the globular proteins. When we say fibrous, these are insoluble. When we say globular, these are soluble. Usually, fibrous, it possesses a mechanical strength, structural components, and for movement. For the globular proteins, it is for transport, for the regulatory and enzymatic actions. In general, four types of interactions they are observed as in the disulfide bonding, electrostatic interactions, the hydrogen bonding, and the hydrophobic interactions. So this is an example of a tertiary structure proteins. So this is your fibrous protein, and this one is your globular protein. Another is your four types of interactions. We have the disulfide bond. The disulfide bond, it is a covalent bonding. That means it's strong and it between two cysteine groups and the strongest of the 3 degrees in its 3 degrees bonds. The links chain usually um, connect and cause chains to twist and bend. So this is your disulfide bond. So in this illustration, we can see the disulfide bond. Okay, these are the disulfide bonds. Another interaction is that your electrostatic interactions. Usually, it is an ionic interaction or the salt linkage. The salt usually bridge between charged side chains of acidic and basic amino acids. Okay? Another, we have the hydrogen bonding interaction. This usually bonds between polar, acidic, or basic R groups. For hydrogen bonding to occur, the hydrogen must be attached to oxygen, to nitrogen, or in a fluorine. Another, we have hydrophobic interactions. This is usually between the non-polar side chains. So this is the hydrophobic interaction. Hydrophobic, that means it hates water. It is usually afraid of water. So these are the examples of your four types of interactions. So this is primary structure to illustrate when we compare it in the cord of the telephone. We have the primary structure, we have the secondary structure, and the tertiary structure. Lastly, we have quaternary structure of proteins. Quaternary structure of proteins refers to the organization among the various polypeptide chains in a multimeric protein. Usually, they has the highest level of protein organization, your quaternary structure. Present only in proteins that have two or more polypeptide chains, and the subunits are generally independent of each other and are not covalently bonded. The proteins with quaternary structure are often referred to as the oligomeric proteins and contain even number of subunits. So, in summary, on the structures of proteins, we have the primary, the secondary, tertiary, and quaternary. So when we say primary structure, the sequence of amino acids present in a protein's peptide chains or chain. Okay, so here another, we have the secondary structure that are regularly repeating ordered spatial arrangement of amino acids near each other in the protein chain, which results them from hydrogen bonding between carbonyl oxygen atoms and amino hydrogen atoms. This leads in the formation of alpha helix and your beta pleated helix. Your alpha helix, hydrogen bonds between every fourth amino acids, and your beta pleated helix, the hydrogen bonds between two side by side chains or a single chain that is folded back on itself. And then if it is anti-parallel, it is more stable compared to a parallel position. Next, we have the tertiary structure. Tertiary structure has three bondings or three interactions. We have disulfide bonds, electrostatic interactions, we have hydrogen bonding, and the hydrophobic interactions. Lastly, the most complex structure of the protein is your quaternary protein. Your quaternary protein structure usually have their interactions, the electrostatic interactions, hydrogen bonding, and hydrophobic interactions. This is usually three-dimensional shape of a protein that consists of two or more independent peptide chains is your quaternary structure. So see the differences of primary, secondary, tertiary, and the quaternary structure of proteins. Now, as what I've mentioned in the previous slides, we have the fibrous protein, which is the alpha keratin that provides protective coating for organs. So major protein consistent of hair, constituent of hair, feather, nails, horns, and turtle shells. So mainly made up of hydrophobic amino acid residues, and the hardness of keratin depends on the sulfur or disulfide bonds. Okay, more disulfide bonds make nail and bones hard and hair brittle. Another is your fibrous protein. Example is your collagen. Mostly abundant proteins in humans, about 30% of total body protein, and the major structural material in tendons, ligaments, blood vessels, and skin. And your fibrous protein and the collagen is organic component of our bones and teeth, and predominantly structure is a triple helix. Rich in proline up to 20%, which is important to maintain the structure. So as what you can observe here, it is fibrous because it has a structure that the string structures or hair-like structures. This is usually needed for the strength of our proteins. Another here is your globular proteins. One example is your myoglobin. Your myoglobin is a globular protein that the oxygen storage molecule in the muscle, especially in skeletal muscles. The monomer is a single peptide chain with one heme unit and binds one oxygen molecule. Your myoglobin has a higher affinity for oxygen than the hemoglobin. So the oxygen stored in myoglobin molecule serves as a reserve oxygen source for working muscles. Another here is your hemoglobin. Your hemoglobin is an oxygen carrier in blood and usually transport oxygen from lungs to tissues. It is said to be a tetramer, that means four polypeptide chains, and each subunit has a heme group and can transport up to four oxygen molecules at a time, and the iron atom in heme interacts with the oxygen. Next is your glycoproteins. Glycoproteins are conjugated proteins with carbohydrates linked to them. So usually many of plasma membrane proteins are glycoproteins, and these are important in their blood group markers of the ABO system, and usually collagen and immunoglobulins are glycoproteins. Collagen or glycoprotein, most abundant protein in human body, which is 30% of the total body protein. Usually it is triple helix structure and rich in hydroxyproline and 5-hydroxylysine derivatives. Some lysines are linked to glucose, galactose, and their disaccharides that usually help in the aggregation of collagen fibrils. Next is your immunoglobulins. Immunoglobulins are glycoproteins produced as a protective response to the invasion of microorganisms or foreign molecules that are usually enters in our body. So these are present in antibodies against the antigens. So immunoglobulin bonding to an antigen via variable region of an immunoglobulin. Okay, so this is the variable region. Usually through hydrophobic interactions, the dipole-dipole interactions in hydrogen bonds. The carbohydrate molecules attached to heavy chains, okay, the heavy chains here, this one, the H, aid in the determining the destinations of immunoglobulins in the tissue. So this is your immunoglobulin molecule that bonds with your antigen that makes responsible for the antibody, or I mean immune response of the body when the body labeled it as intruder or labeled it as foreign material or foreign molecule that enters the body. Another, there is a conjugation of protein and lipids. This is your lipoproteins. And the major function of lipoproteins is to help suspend lipids and transport them through the bloodstream. So we have four major classes of plasma proteins: the chylomicrons, the VLDL are the very low density lipoproteins, the LDL are the low density lipoproteins, and HDL, which is high density proteins. So we have the chylomicrons. This usually transports dietary triacylglycerols from intestines to liver and to adipose tissues. The VLDL is for the transport of triacylglycerol synthesized in the liver to the adipose tissue. The LDL, or the low-density lipoproteins, that transports cholesterol synthesized in the liver to the cells throughout the body. And your high-density lipoproteins, or HDL, that collect excess cholesterol from body tissues and transport it back to the liver for degradation to bile acids. And these are how important our lipoproteins in the metabolism. Now, we talked a lot on the formation of the proteins, on the structures of proteins, and how do we destroy proteins. And this is called as your protein denaturation. Partial or complete disorganization of proteins' tertiary structure. This is your protein denaturation. Like, for example, we have cooking food. When we cook food, it denatures the protein but does not change protein nutritional value. Another example is coagulation. This is actually the precipitation or the denaturation of proteins. Like, for example, in egg white. The egg white is a concentrated solution of a protein albumin. It forms a jelly when heated because the albumin is denatured. And what happens to the appearance? It turns white. In cooking, it denatures protein, as what I've mentioned earlier. It makes easy for the enzymes in our body to hydrolyze or digest the proteins because heat denatures the bonds or denatures the proteins. That means it destroys the bonding between the structures of that protein. Cooking also kills microorganisms by denaturation of the protein. And for example, we have fever, that means greater than 104 degrees Fahrenheit, which is 40. The critical enzymes, which is the critical enzymes of the body, starts getting denatured. So this is an example of protein denaturation wherein we add reducing agents. It destroys the disulfide bridges, causing to sulfhydryl groups, and it will destroy the bonds. Also, this your sulfhydryl groups will be oxidized by an oxidizing agent and reform disulfide bridges. Okay? Here are selected physical and chemical denaturing agents. Like we have the heat, which disrupts the hydrogen bonds by making molecules vibrate too violently, produces coagulation as in the frying of an egg. We have the microwave radiation that causes violent vibrations of the molecules that disrupt hydrogen bonds. Another, we have the ultraviolet radiation, the same also with heat action. Violent whipping or shaking causes molecules in globular shapes to extend to longer length, which then entangles. So example is beating of white egg white into meringue. Next, we have detergents. It actually affects the R group interactions. Another denaturing agent is the organic solvents. Like, for example, we have the ethanol and 2-propanol and acetone. Usually, it interferes with the R group because these solvents also can form hydrogen bonds. And it quickly denatures protein in bacteria. That is why we kill bacteria at the 70% uh, ethanol in in disinfecting the surfaces. So strong acids and bases also disrupts hydrogen bonds and salt bridges, and prolonged action leads to actual hydrolysis of peptide bonds. Another denaturing agent is the salt of heavy metals like the mercury, the the metal ions here will combine with the sulfur hydrogen group or sulfhydryl groups and form poisonous salts. Another is the reducing agent that reduce disulfide linkage to produce the SH groups. And that ends the presentation for proteins. If you have questions, you may able to message me in the group chat so that questions may be entertained. Thank you for your kind attention and happy learning.